RAD5 HIV-1 preventive vaccine. Background: safe and effective vaccine for the prevention of human immunodeficit virus type 1, HIV-1. Infection is a global priority. We test the efficacy of a DNA prime recombinant adenovirus type 5 boost DNA RAD5 vaccine regimen in persons at increased risk for HIV-1 infection in the United States. Methods: at 21 sites, we randomly unseat 2,504 men or transgender women who have sex with men to receive the DNA RAD5 vaccine. 1,253 participants, or placebo 1,251 participants. We assessed HIV-1 acquisition from week 28 through month 24. Term week 28 plus infection, viral load set point, mean plasma HIV-1 RNA level 10 to 20 weeks after diagnosis and safety.
Hey dear, let us familiar about swine flu in detail now. Swine flu is also called to be swine influenza, pig influenza, hog flu and pig flu. Technically it is also called and known as H1N1 virus. It is an infection caused by any one of several types of swine influenza viruses. Transmission of the virus from pigs to humans is not uncommon and does not always change to humans, through which results often only in the production of antibodies in the blood. If transmission causes human flu, it is called to be zoonotic swine flu. People with regular exposure to pigs are at increased risk of swine flu infection. Most of the symptoms of swine flu include cough, fever, sore throat, stuffy or runny nose, body aches, headache, chills, fatigue. Top 10 preventive measures. Number 1. Wash your hands frequently and thoroughly. Number 2. Cover your mouth and nose when you sneeze or cough. To avoid contamination of your hands, cough into a tissue or other handy cloth. Number 3. If one has swine flu, it is transmitted to others starting about 24 hours before you develop symptoms and ending about 7 days later. Number 4. If you do have swine flu, H1N1 flu, you can give it to others starting about 24 hours before you develop symptoms and ending about 7 days later. Number 5. If a member of your household has swine flu, designate only one household member to be responsible for the ill person's personal care.
The 6-plasmid DNA vaccine expressing clade B, GAC, POL, and NEF and F proteins from clades A, B, and C was administered at weeks 0, 4, and 8. The RAD5 vector boost expressing clade B, GAC, POL fusion protein, and F glycoproteins from clades A, B, and C was administered at week 24.
Swine flu is a respiratory disease caused by a relatively new strain of influenza virus known as H1N1. It is a contagious viral infection transmitted by inhalation or ingestion of infected droplets, such as from people sneezing or coughing. The H1N1 virus has multiple parts. Inside the virus is genetic material that carries a blueprint for building copies of the virus. The virus has an envelope with spikes of proteins referred to as H and N protein, which give rise to the naming convention for the virus. Swine flu typically infects cells in the respiratory system first. such as the nose, throat, or lungs. The H spike on the virus attaches to receptors on the surface of the healthy cell membrane. This allows the virus to penetrate the cell. As it does so, the body forms a membrane around the virus molecule. The virus then travels towards the cell's command center, the nucleus, and opens up to release its genetic content inside the cell's cytoplasm. The viral genetic content enters the nucleus, where it uses the cell's own machinery to print copies of itself. Some of these copies travel out of the nucleus, back into the cytoplasm. There they attach to ribosomes. Ribosomes are like factories that produce parts based on a blueprint. The viral blueprint causes ribosomes to manufacture proteins, like the H and N spikes, that are needed to create new virus molecules. Now all components needed to create a new virus assemble below the cell membrane, and using the cell's own surface membrane to create the viral envelope, form into a new virus. In this way, a single cell can produce millions of viruses.
Results. In April 2013, the Data and Safety Monitoring Board recommended halting vaccination for lack of efficacy. The primary analysis showed that week 28 plus infection had been diagnosed in 27 participants in the vaccine group and 21 in the placebo group, with mean viral load set points of 4.46 and 4.47 HIV-1 RNA log 10 copies per milliliter respectively. Analysis of all infection during the study period also showed lack of vaccine efficacy. The vaccine regimen had an acceptable side effect profile.
What is going to kill you? Heart disease, cancer, a car accident? Most of us worry about things we can't control, like war, terrorism, the tragic earthquake that just occurred in Haiti. But what really threatens humanity? A few years ago, Professor Václav Smil tried to calculate the probability of sudden disasters large enough to change history. He called these massively fatal discontinuities, meaning that they could kill up to 100 million people in the next 50 years. He looked at the odds of another world war, of a massive volcanic eruption, even of an asteroid hitting the Earth. But he placed the likelihood of one such event above all others at close to 100%, and that is a severe flu pandemic. Now, you might think of flu as just a really bad cold, but it can be a death sentence. Every year, 36,000 people in the United States die of seasonal flu. In the developing world, the data is much sketchier, but the death toll is almost certainly higher. Now, the problem is that this virus occasionally mutates so dramatically it essentially is a new virus, and then we get a pandemic. In 1918, a new virus appeared that killed some 50 to 100 million people. It spread like wildfire, and some died within hours of developing symptoms. Are we safer today? Well, we seem to have dodged the deadly pandemic this year that most of us feared, but this threat could reappear at any time. The good news is that we're at a moment in time when science, technology, globalization is converging to create an unprecedented possibility. The possibility to make history by preventing infectious diseases that still account for 1/5 of all deaths and countless misery on Earth. We can't do this. We're already preventing millions of deaths with existing vaccines, and if we get these to more people, we can certainly save more lives. But with new or better vaccines for malaria, TB, HIV, pneumonia, diarrhea, flu, we could end suffering that has been on the earth since the beginning of time.
Conclusions. The DNA RAD5 vaccine regimen did not reduce either the rate of HIV-1 acquisition or the viral load set point in the population study.
From the time that HIV was first discovered to be the virus that causes AIDS back in 1983, scientists have been working on developing a vaccine. Here we are, more than 30 years later. So why don't we have a vaccine yet? Oh, and by the way, what is a vaccine, and how does it work to prevent an infectious disease? Unlike antiviral medicines used to treat infections, vaccines work by training a person's immune system before exposure to a virus, so that the immune system can fight off the virus and prevent that person from becoming infected. HIV presents some unique challenges to developing a vaccine. First, HIV attacks the very immune cells that are sent by the body to kill it off, and inserts its genes into these cells. After that happens, the only way to get rid of HIV is to kill off the infected cells. That leads to the second challenge, which is that HIV-infected cells can hide out and remain dormant or asleep, making it difficult or impossible for other killer immune cells to find it. Thirdly, every time HIV reproduces, it makes a lot of genetic mistakes, and unlike many living things, including humans, that have certain systems in place to correct these mistakes, HIV is a sloppy virus that doesn't fix itself. So, HIV changes over time, and it has been difficult to find a vaccine that will protect against all of these changes. So how do we propose to build a vaccine? First, you should know that there is never any live or killed HIV in a vaccine, which is a strategy used by some other vaccines against less dangerous viruses. Also, no one participating in a vaccine study is ever exposed to HIV as part of the study. Now that we have that out of the way, the goal of an ideal HIV vaccine is to activate both antibodies and T cells, so that the immune system is ready to fight off HIV. Antibodies are proteins that attach to HIV before HIV can infect a cell, thereby preventing infection. An example of how this works is the polio vaccine. After you get a polio vaccine, you develop antibodies against polio that are ready to attack if you ever come in contact with the polio virus. We'd like to develop an HIV vaccine that develops antibodies that would attack the many strains of HIV that exist. Another strategy is to train T cells to recognize and fight off HIV, so that when HIV enters, the T cells can kill the virus. All HIV vaccines are made with synthetic, manufactured imitations of parts of HIV, a strategy that can never cause infection, but can train the immune system to fight off HIV. The most successful HIV vaccine strategy so far is a combination of two HIV vaccines tested in Thailand that prevented 31% of new infections. This vaccine isn't licensed, but scientists are using information from this study to make more powerful and long-lasting vaccines that can be used for populations around the world. One thing to know is that the most common way to diagnose actual HIV infection is to test for HIV antibodies. A person who becomes HIV infected generates antibodies against HIV, but because they are only developed weeks after infection, they can't prevent infection. These antibodies can keep HIV in check for a while, but eventually HIV comes out of hiding and causes AIDS, unless a person is treated with antiretrovirals, HIV medicines. Measuring HIV antibodies is a relatively cheap and easy way to test whether someone has been HIV infected. But the test doesn't usually turn positive until several weeks or several months after infection, because that's the length of time it takes to develop antibodies without a vaccine. Some people who get a vaccine will also be antibody positive because they have vaccine-induced antibodies, also called